I love those motivating speeches. I should hire you to keep the morale up in the group when one of our models fails. So I will tell you about an approach which, as Rolf said, I hope will lead to a revolution in psychiatry. And I've termed this approach Translational Neuromodeling, and this talk is about what this means. So in medicine, many of the diseases we deal with are so-called spectrum diseases. They're not unitary things, but rather they arise from the interaction of many genes, each of which conveys a little bit of risk. And unfortunately, these genes tend to interact quite strongly with a lot of, and largely unknown, environmental factors such as nutrition, infection, social stress, and so on, which means that across patients that show similar or even the same symptoms, we are in fact dealing with very different hidden disease mechanisms. And that creates a huge problem for the doctor because he or she has to deal with an enormous variability in treatment response and outcome, even when the symptoms are the same. Now, one disease that you all know is hypertension, high blood pressure. That is a spectrum disease. It has a diverse genetic basis. It has very different underlying disease mechanisms. But say 100 years ago or so, doctors would treat this as one disease. Every patient with high blood pressure would be treated in exactly the same way and classified under the same label. It took us many decades to develop diagnostic tests that were objective, quantitative, physiologically interpretable, and I should have added non-invasive. And thanks to these tests, we are in a position these days to identify for most of our patients very precisely what the underlying problem is, and we can treat people individually, we can treat causally. Now, let's compare that to psychiatry. Let's just take one example, but it's quite representative. Say schizophrenia. Like hypertension, it's a spectrum disease with a diverse genetic basis, gene-environment interactions, and probably very different pathophysiological mechanisms across patients. But unlike hypertension, we don't have a single diagnostic test. Nothing. Absolutely zero. All we do is we tick boxes on lists of symptoms, and due to the lack of mechanistically interpretable tests, we do nothing but trial and error treatment. We prescribe one drug, wait, see whether it works, prescribe another, wait, see whether it works, and so on, to the sufferance of the patients and their relatives, and it's very cost inefficient too. Now, my vision is to change that, and basically do for psychiatry something very similar to what people have been doing over the past 100 years or so for many other diseases, and that is to establish objective diagnostic tests that are mechanistically interpretable. Now, we've already learned that genetics will not help us because of the strong interaction with the environment. We need to find something else, and that something else, from my perspective, is to construct computational models, mathematical models of brain function that we can fit to brain activity data from individual subjects. And if those models are good, then hopefully they will enable us to obtain quantitative and mechanistically interpretable diagnostics, and hopefully, at some point, I can't show that yet, but I have some hints, hopefully also individualized treatment recommendations. This is my version, my mathematical version of personalized medicine or personalized psychiatry that you read about in newspapers these days. So, what I will show you in all brevity is how we develop models, what these models are. I will briefly touch on why it's so important to validate them in animal studies and in human pharmacological studies, and finally, to validate them in the clinical context and translate them to applications. So let's start with the models. Before I mention the models, I should point out that the measurements that we can obtain from the human brain in a non-invasive fashion are basically of two sorts. There is something called fMRI, functional magnetic resonance imaging, where we obtain a measure of blood oxygenation and how it changes as a downstream consequence of neuronal activity changes. This has high spatial resolution, relatively poor temporal resolution. The other sort of technique is electrophysiological techniques such as EEG or MEG, where we measure at the scalp level electrical or magnetic potentials with very high temporal precision but low spatial precision. So given such measurements, you could ask, how on earth are we going to say something about, say, synaptic processes which are meaningful for psychiatric diseases? How can we get from that macroscopic coarse level of large neuronal population activity to micro processes? Well, the answer is we build a model that tells us how any given neuronal state would be translated into one of the measurements that we could obtain. If we have that mathematical description, if it is sound, we can turn the whole thing around and say, given the particular measurement I have obtained, what is the most likely underlying neuronal state? That is the technique that has been developed about 10 years ago by Carl Friston at London, and it's been called dynamic causal modeling. So you can see DCM a little bit like, you could call it a mathematical microscope. We're trying to go from that macroscopic resolution of potentials measured at the scalp level down to microstates, even down to the level of ion channels. I will show an example of that later on. Now, here's an example of what it could look like for fMRI. In this example, you might be interested in getting a measure of synaptic plasticity, in other words, how synapses change their strength. This is one of the key dimensions of disease in psychiatry. And you might be interested to, for example, look at an architecture like this one, where, say, this blue synaptic input here has plastic properties that depends on another input, which, for example, could be a dopaminergic signal from the midbrain. And we can now obtain measures using fMRI that look relatively coarse and washed out. And given an appropriate model, we can infer on the most likely underlying state, neuronal state, under the mathematical constraints imposed by the model, and get quantitative estimates, numbers, of the synaptic plasticity underlying our signals. Now, physiology, however, is not quite good enough. There could be all sorts of reasons why we might observe a change in synaptic coupling. It may not necessarily have to do with synaptic plasticity. In order to constrain our estimates and get more precise and robust measures, we need something else. We need constraints from the theory of information processing. In other words, why does the brain use synaptic plasticity? And from computational neuroscience, there is a strong notion that has emerged over the past decade or so. And it says that basically everything that the brain does, in functional terms, is to do just one thing: reduce prediction error. Every unit of the brain, from the single neuron to the circuit to the whole brain, does nothing but predicting the imminent future and changing its structure online in order to improve its predictions in the future. And it does so by utilizing the degree of prediction error and informing the deployment of plasticity. In other words, the amount of synaptic plasticity that ensues during learning is some function of the prediction error that you generate online. And we have known for quite some time, in fact going back to the 80s, how you would build circuits using very simple equations that can do this sort of learning. Now we can take these long-standing insights and transcribe them into a DCM, which we can now fit to the brain data from our patients. That could look like this. We can apply that, and we shall see that in action now, using an experiment that one of my students, Hanneke and Ouden, did a couple of years ago. There's a very simple experiment, healthy volunteers. Subjects lie in the scanner. They're being exposed to a series of high or low tones. These tones predict, with a certain probability, whether 150 milliseconds later a face or a house will be shown. Your task is very simple. You just have to press a button as quickly as possible and say, this is a face or this is a house. To do that really quickly, you have to learn two things. You have to learn the current probability by which these auditory cues predict the upcoming visual stimuli. And, because we are mean experimenters, we keep changing those probabilities, you have to learn the change in the world, the probabilistic structure of the world, how it changes, the volatility of your environment. Now, to describe that, we need a model that describes how a human subject would optimally learn that and produce online estimates and, indeed, prediction errors. And one way of doing that is going back to a very old theorem in probability theory that goes back to Thomas Bayes, a man of the Church of England, who lived and became famous without ever publishing a paper. But his theorem is one way of describing how you would optimally learn about the world. In this case, we now need to invoke a hierarchical version of his theorem, where we learn about the sequence of sensory stimuli, how they arise from events in the world, how these events in the world are probabilistically coupled in time, and how this coupling changes as the world is changing. So we can apply this model, and I can tell you without showing it that describes human behavior very nicely. But we can now apply it to the brain data and ask, where in the brain is there activity that encodes these computations that the model prescribes. And we find a structure that has been found again and again, showing prediction error activity, namely the so-called putamen, a little structure which I've circled in red here, in the middle of your brain, part of the so-called basal ganglia. So if you now remember that prediction errors are a sort of teaching signal for synaptic plasticity during learning, then a question arises, an intriguing possibility, namely, could it be that this structure is something like a master structure in the brain that controls, in a trial-by-trial fashion, the strength of connections elsewhere that are needed to do a task? So we took up this possibility and built a little model. And this model is really simple. This model basically describes how visual information, in our case the face and how stimuli, are being fed via synaptic connections to a structure called the premotor cortex, which is a structure that plans your motor response, in this case the button press. And this model says or describes how the prediction error activity of the putamen regulates the strength of those information channels, if you will, dynamically to ensure that the system optimally learns from its errors and predicts the next stimuli best. Very simple model, but now, note, we can fit this to the data from individual patients, or subjects in this case. We could get numbers of how much a particular individual learns from prediction errors and how that is being transcribed into synaptic plasticity. In our case, this was a highly consistent and significant effect across subjects, and at the moment, we're conducting pharmacological and genetic studies to find out what underlies this particular form of plasticity that our model tells us exists. And hopefully, this will then progress to clinical studies in the very near future. Now, I want to show you one more thing in this regard, and that is even without having a model of the physiology, just being able to describe quantitatively how people learn and learn from their errors can be diagnostically interesting. I will show you two subjects here. One is a healthy control, and the other one is a prodromal schizophrenic subject, which means this is a person who's not ill yet, but who shows subtle signs of psychosis, who has a high risk because they're family members who are psychotic, but this person is fully functional and he can do pretty much any task. So this is a simple reward learning task where they learn to predict upcoming monetary rewards. And when you look at their behavioral responses, and I show two distributions here in blue and in red for the two subjects in this case, they're pretty much indistinguishable. The healthy control and the prodromal patient are not different when you look at the statistics of their responses. But when you use the model to quantitatively describe learning at different levels, you find a very dramatic difference. Both are capable of learning the trial-by-trial probabilities, but only the healthy subject can learn that there is something in the world that changes, that the probabilistic structure evolves over time. The prodromal schizophrenic patient doesn't do that. He compensates in other ways, but there is a very clear difference. And now we have numbers describing that difference and describing the nature of the learning. So this may all look all very nice, but we have to remember that with mathematics we can do pretty much everything. And we need to remind ourselves that models are caricatures of the real world. Every model is wrong. Some are useful. We have to check whether ours are useful in order to solve certain problems. Now, I want to show you just one study of validation, very recent. This is work done by my colleague Rosalind Moran at London in collaboration with a number of friends at London. And this is a study with healthy volunteers receiving drugs, placebo or L-Dopa, which is a dopaminergic drug that is being used for Parkinson's treatment. And we know from previous animal studies precisely what happens to particular cells in the prefrontal cortex when they're being exposed to dopamine, or more precisely, we know the changes at the level of ion channels that happen under the influence of these drugs. Now we can do these experiments in humans. We can measure activity using MEG at the surface level from the scalp, completely non-invasively. We can apply a model and ask, does the model recognize those changes in ion channel function? And the answer is yes. It replicates precisely what's been shown before in animal studies. And what is more, the changes in those ion channel conductances correlate with the behavior changes that are induced by the drug. So now we have a functional assay of the hidden processes at an ion channel level that predicts how subjects will respond to a drug in terms of performing a task. In this case, this was working memory. And you can now imagine that if we can replicate this result, we don't know yet, if this is robust and holds up, we're getting very close to a non-invasive functional assay that can be used for predicting treatment responses in patients. But we don't know yet. Time will tell whether this is robust. So let me show you one thing that is quite robust, although perhaps it's not quite as useful as the thing I showed you before. And this is now a clinical example. And this is a question which we addressed merely because we thought it was a nice proof of concept study. It was only recently that I realized, talking to clinicians, that it could actually be also become useful for clinical purposes. And this is the question. If we want to validate a model, we have to benchmark it against something we know. Something that we do know in some patients is whether or not there is a lesion. So here we took patients who had a lesion, and that lesion made them aphasic. They had speech problems. Now we asked, given that they have this lesion in some part of the brain, can we obtain a measure from the healthy part of the brain parts that haven't been damaged by the stroke? And in this case, we looked at a network in the auditory cortex, and we were asking, can we detect, using a model, whether or not there is a hidden lesion elsewhere, a remote lesion? And in fact, this is the sort of question that is being asked in the clinics when you deal with, say, multiple sclerosis or with blast injuries, diffuse traumatic brain injuries of soldiers, for example. So here we had patients where we knew whether or not a lesion was present, and we were hoping to see that as a consequence of that lesion, we would find changes in the synaptic coupling in the network that was not touched by the lesion. So we obtained fMRI data from our patients and healthy controls while they were listening passively to speech in the scanner, using fMRI. And now we built a model, very simple model of the auditory cortex, interacting auditory regions, thalamus, primary auditory cortex, planum temporale. We fit that model for every subject, and we now decide on the basis of our parameter estimates, is there a lesion hidden remotely or not? And the answer is, with almost 100% accuracy, we can decide that on a case-by-case basis. You can take any other tool from contemporary statistics. I'll just show 3 here. We tested another 15 or so. They perform far, far worse. They don't really get anywhere beyond 70%. And visually, you can see, you can take the most discriminative data points. The two groups are not really well separated, but the model really pulls them apart very, very nicely. It can find those subgroups. And remember, we're looking at data from the healthy part of the brain. Yeah? So this may illustrate the power of those models. If you get a good model, of course, that's always contingent on your model. But what I have shown here can be summarized in one slide. What we are trying to do with this approach, which I call translational neuromodeling, harnessing the power of computational modeling, bringing them into the clinical application domain, we hope that we will establish non-invasive assays of human brain pathophysiology that will enable quantitative diagnostics and hopefully at some point individualized treatment. And with that, I would like to thank my collaborators. We've talked of collective intelligence before. This is not just my work. Many friends have done this together with me. My group at Zurich, my friends at London, at Cologne, colleagues at Zurich, Brisbane, New York, Hull. My thanks to them and my thanks to you for your attention.